Hey, welcome back. Well, you're right. We are now in the last chapter, which is on genetics and heredity. And we're going to start with a review of the simple Mendelian genetics that you have learned in biology. So, this is only made up of a very short slide. This is part one. It's only made up of 16 slides, but let's do it now. So the Mendelian genetics was given to us by Gregor Mendel. He's a monk. He was a monk, and pretty much he wanted to do so much with science. He loves biology so much, but according to stories, he did not really make it so well in his master's degree when he was taking his master's degree. So he ended up just doing the works of the things that he loved in horticulture, which is actually the science of raising plants. And he did it like at the back of the monastery where he was a priest at that time. And over the years, well, the abbot of the monastery was very supportive of pretty much what he wanted to do until the abbot died and he became the abbot of the monastery and he continued on. This is more than 10 years. As a matter of fact, um, when he died, he already had made the book. And one of his students pretty much was the one who presented his book to the scientific community because his book was pretty much like not recognized. He was not recognized. He did not have a master's degree at that time, was very important. And only had an undergrad, and his grades were not really that bad, not good, rather. And so one of his students and one of his supporters, actually, who had a PhD, was pretty much the one who presented. This is already after he died. Presented his theories on heredity, on genetics. And this is actually at that time when nobody even knew anything about the DNA, the genes itself. So let's start with looking at some terminologies in genetics. For instance, chromosomes and genes. Now, the complete complement of an organism's DNA is actually the human genome, which consists of about 3 billion base pairs organized into 23 pairs of chromosomes only within the nucleus of each of our cells, plus a small circular DNA molecule in the mitochondria. So we have extra DNA that is actually like in every cell of our body. Besides the 23, we have what you call the mitochondrial DNA. And a typical human cell has a diploid set of 23 chromosomes, a total of 46 with one set inherited from each of the parents. So you inherited your 23 from your dad, and then you inherited another 23 from your mom when the egg fertilized, when the sperm fertilized the egg. So the chromosomes has our own entire chromosome says is um, more than 10 billion base pairs, but actually our genes is actually something like the likes of about 30,000 genes. And you could just pretty much like mix and match, and we have 30,000 characters that you could actually come up with in that particular 30,000 genes. So the cellular DNA actually is organized in genes. Now we call them genes, and each of the genes have their specific place on the chromosome. And if you're going to take a look at the chromosome, and right here you have a chromosome. Let me just check on that. Right here you have a chromosome, and you have the exon, introns, and then you have exons here. Exons stand for expressed uh, genes. So these are parts of the DNA that become, which we call genes, that become the characteristic traits. Their expression is um, actually part of our trait. The introns, these ones here, are intervening sequences. They're garbage, but they don't really express for anything much. But we have accumulated them over the years of evolution, and pretty much we learn about our past through the study of introns. They are not really garbage, they become part of our signature, and geneticists study. There are special lines of geneticists that study only the introns, and they realize that the introns significantly will say something about our past. So they do have specific places on the chromosomes, the genes, which we call the locus. So who was Gregor Mendel again? Oh, well, now we call him, the father of genetics. That gentleman then, whose work was not really even accepted because he did not have clout, is now considered to be the father of genetics. And really, truly, he was a genius because all of the things that he actually worked on for more than 10 years, and he wrote it down in a book, pretty much came in and as a part of his observation only, and his experiments in the piece, and pretty much um, uh, it was actually not considered to be like um, good by the people then because he did not have any clout, but he really did really very good work. So uh, So this is actually in the decade of 1860. It's in an Austrian monastery, and the monk's name was Gregor Mendel, and spent his spare time breeding pea plants. He did not, he did this over and over and over again, and noticed that there's a pattern of inheritance from one set of pea plants to the next, from one generation to the next. And carefully he analyzes pea plant numbers, and he discovered three laws of inheritance, which actually he wrote in his book, which is really very credible. And Mendel's laws are as follows. He has the law of dominance, the law of segregation, and the law of independent assortment, which we will discuss in in simple terms today. So in his work, the word chromosomes pretty much did not come out yet, because chromosomes and genes are nowhere to be found in the vocabulary of scientists in the 1860s. The role of these things in relation to inheritance and heredity has not been discovered yet, but he used his own language. So what makes Mendel's contribution so impressive is that he described the basic patterns of inheritance even before the mechanisms for inheritance, which is actually our genes, was even discovered. Okay, so a little more of the terminology. Um, the law of dominance is the law of segregation, and the law of independent assortment is actually the laws that we're going to concentrate on. This is a very short discussion. Then we are, together with that, we're going to be looking at a little bit more of genetic terminology, which is going to be part of your exam. So, genetic terminology. A genotype is actually the genes of an organism. All of your genes will be part of your genotype. The phenotype will be the traits of the organisms that are expressed. Okay, because you have these genes as part of your genotype, then the expression of this genotype will be your phenotypes. Then you have alleles. An allele is actually the variations of a gene. And they're actually represented by letters for the different types of alleles, depending on what allele or character we're talking about. like a capital of the an uppercase letter and a lowercase letter, both uppercase letters or both lowercase letters. These are actually how we represent an allele. And when you say homozygous, you are referring to a pair of identical allele for a character that would pretty much be represented by the same uppercase or the same lowercase allele. They are homozygous or purebred. Then you have heterozygous. These are two different alleles of a gene, which could be an uppercase and a lowercase. So a character. A character is a heritable feature. For example, a color of hair, color of the skin, color of eyes, okay? Or, are your fingers bent or are your fingers straight? Something like that. And a trait is a variant of a character. For instance, the color of eyes, okay, is actually like a character, and then the color blue is a trait. Color green is a trait. Now, true bred, okay, pretty much refers to all of the offsprings of the same variety. Then hybridization is when you cross two different true breds, okay? And we label the different generations pretty much of a cross when you are going to be crossing parents, okay? As for instance, the P generation being the parents, and you have the F1 generation, which will be the supposedly the 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 progeny of the P generation, which means the uh, the result of the crossing of the P generation will be the F1, the first filial generation. In humans, we call this the P generation being the parents, the F1 generation being the children, and then we have the F2 generation being the grandchildren of the P of the parents. Okay, so this is actually the second filial generation. And so who will be the parents of the F2 generation? Well, could be the first, the F1, okay? So there are more of genetics. Again, terminology, we have your dominant and recessive. Let's take a look at how we actually use them. Um, a homozygous genotype means that the genotype is actually purebred, which means the alleles are actually both represented as, let's say here, both lowercase or both uppercase. Lowercase meaning recessive, and both uppercase meaning it's dominant. Heterozygous is actually a hybrid. At least one, there's going to be one dominant allele in the genotype. For instance, you have an uppercase R and a lowercase r. And we have here a character. The character here is tongue rolling. Is an example of what we're going to do today. And tongue rolling means you are able to roll your tongue, and it is a dominant phenotype, and this is actually a human trait. And Harry, male, is actually able to roll his tongue and is a dominant trait. And the phenotype of Harry, what's the phenotype you think of Harry? And how would we represent the genotype? The phenotype of Harry is tongue rolling, right? But the genotype of Harry could either be homozygous or heterozygous. Remember that the trait itself is actually a dominant trait. So you can have this, okay, you can have that, or you can have a big R or a small r for Harry. So Hermione pretty much is not able to roll her tongue, and pretty much has a recessive phenotype, which is actually a non-tongue roller. Now, what would be the recessive phenotype? Well, the recessive phenotype would be a small r, small r. But Harry, being a dominant or is a carrier of dominant trait, can have a big R, which means a pure hybrid for the dominant trait, or it could have a heterozygous big R, small r, right? But we put it that way, and so the resulting the possible cross between these two would be a heterozygous tongue roller. Now, this is actually the name of this is actually Harriet. Now, would Harriet be able to roll her tongue? Well, because the tongue roller is a dominant phenotype and she carries a dominant trait, then of course she's able to roll her tongue. Okay, now Mendel's law, the first law of Gregor Mendel that we're going to review will be Mendel's law of dominance. This means that in a cross of parents that are pure for contrasting traits, only one form of the trait will appear in the next generation. Okay, and what will appear, whatever it is that will appear in the next generation, is actually dominant over the other trait. Okay, so here you have a tall pea plant and a short pea plant, and the tall and the short that has been crossed, then all of the first generation, first filial generation, the offsprings, are all going to be tall. That means there's actually one parent, which is actually the one that has been expressed. The trait of that particular parent is dominant over the trait of the other parent. They're going to have offsprings that are hybrid for a trait and will have only the dominant trait in the phenotype. And what's going to happen if you're going to cross this? Well, by crossing this, Gregor Mendel found out that in the first filial generation, if you cross the first filial generation, the second filial generation will actually reappear. It looks like in this first filial generation, the character or the trait of the recessive trait has disappeared. And that's pretty much what his questions were. What happened to the other trait? How come all of the first generation were all like copies of the one parent? Only what happened to the trait of the other parent? And when he crossed this, the first filial generation, he found out that that particular trait would surface. And so this is actually how he coined the principle of dominant traits. Having dominant trait will mask the effect of the recessive trait, but when you cross hybrids of the dominant trait, you will actually express the recessive trait in pretty much a very small percentage, but it will be expressed. Okay, on the last line that we have, if Harry was heterozygous, what will be the possible genotypes for Harriet? Well, the answer is 50% tongue roller and 50% non-tongue roller for Harriet. Mendel's second law is actually the law of segregation. Pretty much the law of segregation brings about the study on alternative versions of the genes, which we call alleles, and they result in variations in inherited characteristic traits. Now, for each character, an organism inherits two alleles, which is actually supposed to be one coming from each of the parents. And the alleles for each character would segregate, they will separate during the formation of gamete. So when they segregate and they form the gametes will be a mixture of the parents. Okay, and this table shows how the genes actually exchange according to Mendelian law of segregation and independent assortment. Now, during meiosis, um, pretty much the different allelic traits that come in is actually a translation of the Mendelian law. During the law of, during the mixture or the mixing, when the cells would prepare to divide, during the mixing of the chromosome would also cause a mixing of the genes, and the genes actually will segregate, which means they separate from one another. Which means if you have two traits, that's actually a yellow pod and a green seed. This yellow pod and green seed, according to Mendelian law of segregation, will not will not be coming together, or will not always be together during the production of gamete. Yellow pods and green seeds will separate or may separate and will actually be um, carried in different offsprings. Okay, that's actually the law of segregation, which means, okay, in human application, if you are blonde and blue eyes, you cannot always expect that your children will also be blonde and blue eyes. You can have a child that's only blonde and would have brown eyes, okay, because being blonde and being blue eyed, these particular traits really do not stay together whenever there's actually crossing over in meiosis. Except for one, there's actually one particular human trait, well, which pretty much is not covered by Mendel's law of segregation. And that's actually being a redhead and having freckles. All redheads always would have freckles, which means regardless of what you do, if you are a redhead, you will always have freckles, because the genes for being a redhead and having freckles are so close together in a chromosome that during meiosis, they are not really um, being separated during meiosis and during the process of crossing over. They always will be going together. Okay, that is actually one of the things that's not covered by Mendel's law of segregation that is in human genetics. And Mendel's law of independent assortment states that alleles for different traits are distributed to sex cells, and that means the offsprings independently of one another, kind of like similar to the law of segregation. Now, remember, guys, Mendel came up with all of these ideas even before we knew about the existence of a DNA and the genes and the chromosomes. He called all of these particular phenotypes traits. Period. They are just nothing to him but just traits. But he was able to eloquently come up with the idea and come up with a discussion, an eloquent discussion of the law of independent assortment and segregation. So let's recap Mendelian law. The law of dominance, okay, in a cross of parents that are pure for contrasting trait, only one form of the trait will appear in the next generation, and that's actually the dominant trait. Offsprings that are hybrid for a trait will have only the dominant trait in the phenotype. The only time that you're going to have the opposite trait is when your trait is actually, your allele is not dominant. The only time that a recessive trait will be manifested as a phenotype is when both parents will hand down the recessive allele to that particular offspring. In the law of segregation, during the formation of gametes, I mean eggs or sperm, the two alleles, which are supposed to be, according to Mendel, hereditary units, are responsible for a trait that are actually separating from each other. Alleles for a trait are then recombined. When they recombine at fertilization, they produce a genotype for the traits of the offspring. Now, the law of independent assortment says that alleles for different traits are distributed to sex cells and the offspring independently of one another, which means if you're blonde and blue eyes, you cannot say that your children will be blonde and blue eyes. Same thing as you are blonde and blue eyes. Most especially if your spouse is actually not blonde and blue eyes. So Let's try to figure out patterns of inheritance using a particular tool which we call the Punnett square. It's actually a tool for diagramming the possible genotypes of offspring. Let's do a Punnett square for the trait of bent little finger, which is in humans a dominant genotype, uh, using the letter B to represent the allele. So it's a dominant. So B meaning bent. So David, which is the baby daddy, okay, has a dominant phenotype. So David here has a bent finger. So what could be David's phenotype? Well, we're going to find out what David's phenotype is later. Tammy, the baby mama, has a recessive phenotype. She has straight finger. There's no picture of Tammy's finger here, but most of us really has already a straight finger, which is actually, because it's not dominant, it's actually recessive, is very, to humans, we have it actually as the more common one. So what is a genotype of Tammy? Well, we're going to look at the genotype of Tammy here also. And that's the genotype of Tammy right there. Small b, small b. Leo, the baby, uh, pretty much carries the dominant phenotype, which is bent finger. If you can see the baby's finger there, tiny finger, he has actually a bent finger. So the baby's genotype would probably be either a big B, which is homozygous for being having bent finger, or a big B, small b, which is heterozygous for a bent finger, because it's a dominant trait. Both homozygous and heterozygous genotype will have a phenotype of bent finger. But there's a hint here, so we can actually create our Punnett square. Baby has an older sister that has a straight finger. So that's going to be like easier to determine now. David's genotype could be, is it a big B or is it a big B, small b? Is he actually a hybrid, or is he a purebred for the trait? Let's find out. If he was a purebred for the trait, that would be big B and big B over here. Let's so take a look at that. You have the mother's genotype right there. If David is big B, let's put big B here, then all of the children will have bent finger. But remember the hint. Baby's older sister has straight fingers. So the father cannot be a homozygous for the dominant trait. So the father, I assume, is a heterozygous. When he's a heterozygous, let's find out if the baby will be there. Baby is has bent finger, that right there, and then the older sister, has straight finger, and that's your straight finger right there, and straight. All right, so far we have discussed simple inheritance and an example of the use of the Punnett square. But of course, genetic is much more complicated than what we have just discussed. We are going to be exploring some more of the simple inheritance that went after, or that was actually studied after the death, and pretty much the presentation of the Mendelian genetics after his death and after the recognition of his book. There are several scientists who went and joined a bandwagon and studied genetics, and in their exploration came up the incomplete dominance, the multiple alleles, the codominance, the polygenic inheritance. There are so many. And so epistasis, so many, but we are only going to be concerned with a few that would pretty much be part of the human genetics. So incomplete dominance. So this is actually beyond Mendelian, okay? Incomplete dominance is a pattern of inheritance where the dominant trait often go uh, beyond what do you call this, pure expression. I mean, I say beyond. It cannot go beyond pure expression because pure expression is just whatever the dominant trait is. Well, this simply means that if you have a dominant trait, okay, that is actually red, and you have a recessive trait which is actually white, uh, the the red cannot pretty much like exert an overpowering dominance over the recessive counterpart, and therefore they come up with a blending, which is actually a pink. Okay, that's actually the incomplete dominance. Actually the incomplete dominance of the red over the white. So uh, there's actually a blend between the phenotypes of both the parents. And when you cross two incomplete dominant traits, then you will come up with a possibility of 25% pure dominant red, 25% of the recessive white, and then two of the blended incomplete dominant traits. An example of this will be your snapdragons. This is the genetics of snapdragons. Then we go again in another concept that went after Gregor Mendel is the principle of multiple alleles, where inheritance actually, where in an inheritance of a trait, you have actually several alleles that are actually involved. And examples of this trait will be the trait for the eye, the character of the eye color, and then the human blood types. These two traits pretty much carry different alleles. For instance, in the eye color, you can have um, black, and then you can have also a green. You put them together, and you can have really like a slew of combination that where you can have different eye colors. And it's because the eye color is actually made up of several alleles. It's not only the allele for green and black. There are there are actually other alleles, but the black color of the eye actually is a dominant trait. The green is actually the super recessive, okay? And because they are made up of several alleles, you put them together and the combination of these several alleles would be blending. Okay, and then you have also the blood typing. Blood typing are also allele, multiple allele. They're multiple allelic, they're polygenic, they're made up of the allele for A and the allele for B, and then together with the allele for A and B, they can create, and then of course the allele for A and B and not having A and B. So we're going to discuss this further when we get into the next part. Okay, right now I'm just giving you that the blood typing is multi-allelic. We're not going to be discussing this fully because this is AMP2. When we get into the blood in AMP2, you are going to pretty much do your own blood typing exercise. But right now, it's just knowing that blood typing or the human blood types is actually multiple allelic. They're made up of more than two alleles, and that's actually the allele for being blood type A and being blood type B. And that's it, guys, for the first part of simple genetics.